Anthelmintic drugs. Infection with the parasitic worms is the most common disease in many tropical and subtropical countries. These parasitic worms firmly hold the intestinal mucosa and continue their reproduction by egg formation. Worms parasitic to human being can be categorized as cestodes, nematodes, and trematodes, that is tapeworms, roundworms, and flukes. Anthelmintics are the agents which are used to destroy or eliminate the parasitic worms from the GIT without causing damage to the host. They act by killing or paralyzing the worms so that such worms could be easily expelled out of gut. Some anthelmintics also impair the egg production process in worms. Depending upon their action, anthelmintic agents are categorized into two types, that is vermifuges and vermicides. Vermifuges means drugs that expel the worms from the body, and vermicides means drugs that kill the worms inside the body. Classification of anthelmintic agents. Anthelmintics are classified based on their chemical structure. They are mainly classified into 5 different classes, that is piperazines, benzimidazoles, amides, phenols, and chlorinated hydrocarbons. Piperazines, for example, diethyl carbamazine citrate. Benzimidazoles, for example, albendazole, mebendazole, and thiabendazole. Amides, for example, niclosamide. Phenols, for example, dichlorophane. And last class, chlorinated hydrocarbons, that is tetrachloroethylene. Piperazines, for example, diethyl carbamazine. It is an orally active agent belonging to piperazine category. It acts by sensitizing the microfilaria so that they become phagocytosed by fixed tissue macrophages. Next class, benzimidazoles, for example, albendazole, mebendazole, and thiabendazole. The main pharmacophore present in all these structures is the benzimidazole nucleus, having two substitutions at the second position and at the fifth position. In case of the albendazole, carbamate functional group is present at the second position. In case of mebendazole, again carbamate functional group is present at the second position. But in case of thiabendazole, thiazole ring is present at the second position, and the thiopropyl group is present at the fifth position in case of albendazole, while the benzoyl group is present at the fifth position in case of the mebendazole, but there is no substitution at the fifth position in case of the thiabendazole. These all are the broad spectrum anthelmintic agents. Albendazoles generally blocks the glucose uptake by larval and adult stages of susceptible parasites due to decreased glycogen stores and decreased formation of the ATP. So parasites get immobilized and dies. Mebendazole inhibit the microtubule synthesis in the nematodes, thus irreversibly impair glucose uptake. As a result, parasites are immobilized. Thiabendazole, it exerts the vermicidal activity and it acts by deforming the worm eggs. Amides, for example, niclosamide. Niclosamide is a salicylanilide derivative and it inhibits the glucose uptake, oxidative phosphorylation, and anaerobic metabolism in the tapeworm. Phenols, for example, dichlorophane, it is an effective in the treatment of tapeworm infections. It detaches worm from the bowel wall and promotes its digestion in the gut. Chlorinated hydrocarbons, for example, tetrachloroethylene. Tetrachloroethylene is an unsaturated chlorinated hydrocarbon. It is not used as an anthelmintic agent presently and is now of historical importance only. Presently, it is used in veterinary practice and in the treatment of fluke infections in humans. Thank you.